The last application is part of the Exabiome project, which is a DOE-funded project on exascale solutions for microbiome analysis, part of DOE's ECP Exascale Computing project. And Exabiome has three different pieces of motivation for the science questions that come up. What happens to the microbes living in the environment after a wildfire? How are microbes involved in carbon cycling? How did the microbes affect disease and growth of various types of crops, such as switchgrass used for biofuel and so on? So you can see there are important scientific questions that really require that you understand the microbial communities that live within soil. And these soil microbiome communities are very complex. They can have thousands of different species in them, and they occur at varying abundances within the sample. So you can have very common species and then very rare species within the sample. And even the low abundant species can sometimes have a significant impact on the behavior of that soil microbiome. For example, whether it's absorbing carbon or whether it is going to produce high quality crops or whatever. Now, the de novo assembly problem then is to try to understand each of the species that are in one of these samples of soil. And the basic assembly problem, which occurs even for things like the human genome, is to take the raw output of the sequencers, which, by the way, fragment the genome into a number of different pieces, into short pieces, and then put those together in order to construct the complete genome. Now, in the human genome case, we actually have a reference genome because we've assembled that before, multiple times actually. And although there are variations across different humans, much of that is conserved, and so you can just map those back to the original genome. But in de novo assembly, and as is the case in microbial species, in many cases we've never seen them before. There are new species that we've not sequenced before, so we don't have a reference. So the de novo assembly problem is to take the sequencer output and figure out how to put it into a completed genome as the output, or at least large fragments of the genome such that we can find genes and so on in it. And what you see in this little graphic is also the idea that not only do the sequencers sort of shred the genome into these short pieces, but they also insert errors. And they can both insert characters, delete characters, and flip characters. So all of those things are errors that the sequencer has to deal with. And the way that you deal with it in de novo assembly is you read the genome multiple times so that you assume that you have enough good copies of any piece of the genome that you'll be able to reconstruct it. And you can sort of see that here as we've tried to align up the different reads, and that is what the output of the sequencers are called, these substrings. And by having multiple copies, you can resolve what those errors are. So this problem is like putting together a puzzle where you don't actually have the cover. If you had the reference genome, it would be like putting together a puzzle with the cover of the puzzle box. In this case, you're trying to put together the puzzle without knowing what the box looks like. And by the way, somebody's thrown in some extraneous, erroneous pieces, but you have multiple copies of the puzzle. So eventually you'll have one good copy of every piece that you need to complete the puzzle. So in looking at the environmental microbiome now, as I mentioned, you have, say, up to thousands of species that are living together in one of these communities. And you literally scoop up some water or some soil and you send that sample through the sequencer. And so all of those little fragments that the sequencer produces are mixed together in the sample. And so now the problem is really quite different. It's really about putting together a puzzle, a whole bunch of different puzzles, say 1,000 puzzles, with pieces from all of them dumped together on the floor. This analysis of this large-scale environmental microbiome and the use of MetaHitmer for this problem was actually a best paper finalist at SC18, which you can find online. The basic idea, though, and the motivation for having an exascale or at least an HPC assembler is that by combining these very large data sets together and co-assembling them, by assembling them as one big group, then we can actually get better quality output. And I won't read through the details of this slide, but just to give you a sense of sort of the state of the art in these metagenome analysis and assembly problems, a lot of people use something that we call multi-assembly, which is you've got the data in different subsamples, in this particular case from the Twitchell wetlands, samples. It was in 21 pieces, and you can assemble each one of those separately. Now you can imagine that for the common species, the high abundance species, you can get a pretty good assembly out, but for the low abundance species, it's very hard because you don't have enough copies of the puzzle pieces of those low abundance species. And that's really what the right-hand graph is showing and quantifying, is that the number of genomes that you get out and the completeness of those genomes really improves when you do co-assembly, which is this top blue line, compared to multi-assembly, which is just the orange line. Just doing one lane, just doing one of these little boxes over on the left, is the red line, which is not nearly as good as assembling everything together or even assembling everything separately. When you do multi-assembly, you might have to remove duplicate species that you found, or you should do that, and so you can get a little bit better quality out of deduping the multi-assembly, but still not nearly as good as having one big, huge full co-assembly. And in the examples that I gave in the first part of this, this metagenome description of this case study, there are multi-terabyte data sets, including this wetlands data set, 2.5 terabytes. And the data, by the way, blows up by almost an order of magnitude in the middle of the assembly. And so you can see that you can't just buy, say, a single shared memory machine to do a co-assembly of this kind. And most of the other state-of-the-art assemblers are shared memory assemblers, and especially the only production metagenome assemblers besides MetaHitmer are shared memory metagenome assemblers. So what does the genome assembly process look like? Even for a single genome, it has the following really 4 steps in it. It takes the reads as input. It's going to chop them into fixed length pieces, what we call k-mers, and then it's going to use those fixed length k-mers and count them to figure out whether or not there are enough instances of any given fixed length k-mer that's probably a good k-mer or an erroneous k-mer. And if that k-mer only appears once and you have read the genome, say 30 times, then you're pretty sure that is, if you've only got one instance of that k-mer, it's an error. You then take these k-mers and treat them as a graph, and you build connected components with this graph using sort of a parallel depth first search algorithm. Those are the output of that are called contigs, and you take those contigs and then align them back to the original reads, because remember when we turned it into k-mers, we threw out some of the information there, the connectivity between the different k-mers. So you now use the reads to try to connect the contigs together. And this may seem like, well, why are you taking that original data and combining it with these contigs? Well, the idea is that the contigs are very high quality because you filtered out the reads. The reads are not such high quality, but they can tell you where you might connect the contigs together. And so the last step then is to take that alignment. You actually treat this as another graph where the vertices are the contigs and the edges come from those reads, and you use that to try to scaffold and put that genome into a more complete state. So I won't talk about each of these steps in detail, but I'll give you a sense of what the first couple of steps look like so that you can see actually maybe all 3 of the first 3 steps, and to give you a sense of why this is a good fit for UPC++. And on the right-hand side, you can see get an idea of what are the kind of computational patterns that come up in the application. In k-mer analysis, we're going to do histogramming of the k-mers. In contig generation, then we're doing this connected components computation, but the graph is really stored as this distributed hash table of k-mers, where each of the vertices is a k-mer. You then solve this problem called alignment, which is to try to use imperfect string matching, that is, allowing for insertions and deletions and mismatches using something like a Smith-Waterman algorithm to align all of the reads back to all of the contigs. That all-to-all computation is also going to be a distributed memory computation. The local alignments then will be something that can be done in a single processor core. And the scaffolding is also a graph walk, but a much smaller graph because now we're looking at the contigs. So this code, MetaHitmer, was originally written in a combination of UPC, an earlier PGAS language, actually one that's still in use today and is based on C, and then MPI for historical reasons. The histogramming, the k-mer analysis component had been written before some of the others and was written in MPI and then reused in the MetaHitmer implementation. The other parts of it were written in UPC, and but the team has recently rewritten all of this in UPC++. And let me just point out the main authors of this software. The original code was primarily written by Evangelos Georganas, and then Stephen Hoffmeyer and Rob Egan have been really writing the UPC++ version and continue working on it in collaboration with a larger team. And Aydin Bulic actually wrote the original parallel distributed memory K-mer analysis. I'll also mention that code is based on a code from Dan Roxar's group at UC Berkeley called Miraculous, and that is a pun on the K-mer name, so it's Miraculous spelled M-E-R, and that was a shared memory code, and so this was the first distributed memory implementation of this. I didn't really mention yet this iterative loop around the outside of the pipeline here. If you were only assembling a single genome, you might just use the single pass through this pipeline. But when you're assembling a metagenome, because there are these different species that occur at different abundances, the length of the K-mer that you're using is going to have a trade-off in terms of how many K-mers you're going to find and filter out. That is, if you've got a long K-mer, it's very likely that there's an error inside of that K-mer. So you want short K-mers in order to not throw out so much of your genome. But the shorter K-mers then are harder to put together into contiguous pieces, and by using different length of K-mers then and iterating around this it'll allow us to assemble these metagenomes that have different species of different abundances. So the first step is this K-mer analysis, and this just shows you sort of some examples of what the K-mers look like. In this case, we're showing formers, and we're going to parse the reads, extract all of these K-mers. Now, a K-mer length of 4 is ridiculously small, so in practice you would be using something on the order of 21 up to maybe 51, and that's going to blow up the size of your input data set considerably. So you can see how the internal data structures start to become very large. You don't need to keep all of the instances of the K-mers around at once, but you do need to keep track of how many times you've seen a given K-mer. And so this is using both a distributed hash table and then also a Bloom filter. And we'll use a Bloom filter that you may be familiar with, a statistical data structure that is a very compact way of representing, counting a set of things to see whether or not a particular K-mer in this case occurs. And that Bloom filter will filter out most of the singletons, and so we don't have to store them in the output of this particular phase. This graph looks at the performance of the UPC++ implementation with and without the Bloom filter. The Bloom filter is really a memory-saving device, not a performance-saving device. So this is a runtime plot, so down is good. So actually the fastest thing you can do is run the UPC++ K-mer counter. This is really building a big distributed memory histogram out of a hash table, and that is the fastest thing is to do it without the Bloom filter, but the better memory savings then is with the Bloom filter, so we often need to run it with the Bloom filter on, and that is what you see in the red line. The original MPI implementation, which was a bulk synchronous implementation using a set of stages of all-to-all Vs, is shown in the blue line. So you can see that the UPC++ implementation is faster. It also actually saves memory. Part of that is just that we don't have to keep both the MPI runtime and the GasNet runtime communication layers around, and so it's not really necessarily criticism of one runtime or the other. It's just that having one runtime is more efficient than having two. And so that interoperability is nice, but it does cost you in terms of memory utilization. The code is also much simpler, so there's a real productivity advantage here as well, which also makes it easier to maintain. So that's the first step, is we're putting these things into a hash table and then counting them. The next step in assembly is de Bruijn graph construction. And in this case, we take the K-mers that are already stored in a distributed hash table. So they're spread out over the machine based on the hash function. And we walk through a graph that is based on having a common overlap of K-1 characters. So for example, at the example at the bottom, you've got GAT. The AT overlaps with the AT in the next K-mer. And so you can connect that together in a hash table. So this is the contigs that you get out of this, the connected components of that K-mer graph. Now that graph is really represented as a hash table because you've put those K-mers into the hash table, and along with them you store an extension, the left and right extension, that is the character that came before and after that K-mer in the original input read. And so you can hash the K-mer, look them up, find, say, the right extension, append that character to the key, shift the window over, hash that again, and walk through the graph in that manner. So it's really a sort of hash table lookup and repeated over and over again, and every time you're walking through this graph. Most of those accesses are going to be off processor because as soon as you get any number, a reasonable number of processors, let's say 128, 127 out of 128 times, given a sort of general random a hash function with a random distribution, you're going to go off the node. So there's a lot of communication involved, and so having support for very lightweight, low overhead communication is especially important. This just gives you a picture of what one of these hash tables looks like in the global address space. It's turned sideways here so that so it fits on the slide, but you can see that each processor may be parsing its reads, turning them into K-mers. In this case, the picture shows a 3-mer, and the left and right extensions, which are then stored as the values in the hash table along with the key, as the K-mer as the key. And the hash table has a set of buckets that are distributed over the machine. So inserting these into the hash table involves pushing these out. Most of them will be in messages to remote processors or data transfers across the nodes of the processors, just because the hash function is distributing things evenly. And then you can also, to walk the graph, you can look things up in the hash table, attach one of the characters as I've described before, and then look things up, the next key up in the hash table. This type of data structure gets used throughout, and the Bloom filter, by the way, looks somewhat similar, although is a more compressed version of this type of data structure. So you can see that this is going to require fine-grained communication and, by the way, fine-grained locking. I didn't really talk about the fact that as you're walking through the graph, the De Bruijn graph, which is represented as this hash table, that two threads can run into each other because they're actually they're doing a depth-first search on a config that's going to be the same config, and they haven't yet figured that out. So there is some complicated locking in here as well, which is also made easier through the UPC++ primitives. Now, even in UPC++ with the lightweight GasNet communication layer, there's still a big advantage to having a larger message size. Now, this shows the latency, which maybe doesn't make it quite as obvious, but we also see that these latency numbers, although there's some increase as you increase the number of threads, that is, the blue line is a little bit slower than the others because you're probably going further across the machine and things like that. For the most part, you can see the differences in the running time here. But perhaps even more importantly is this bandwidth plot that shows that for small messages, you really get very low bandwidth. And so if you're only sending 64 bytes, which is actually not an unusual size, that might be a typical size for a K-mer, for example. If you're sending them one at a time, that would really be, would put you at a very low fraction of the peak bandwidth of the system. And so one of the optimizations that has been in both versions of MetaHIPmer, and it was just recently implemented in the UPC++ version, is an aggregated store, which is aggregating dynamically the set of operations, the set of K-mers, for example, that are going to go to the same remote processor. So this allows you to send a smaller number of larger messages, a very common optimization technique in MPI, but even in GasNet, when you're talking about only sending 64 bytes, there's still an advantage to getting messages that are somewhat larger. Here's a little bit of the sketch of the interface for the aggregating stores, just to show it uses the distributed objects in UPC++, and it gives you an interface that is fairly convenient in terms of just sort of updating the set of operations, the set of elements the data elements that you're transferring, and then shipping them off to another remote processor that happens automatically. Now, the third phase after you've built this De Bruijn graph is to do, as I described earlier, to align the reads using some kind of a string matching algorithm that allows for the possibility of imperfect matches between the reads back to the contigs that have just been computed using the depth first search. And that is this global sort of alignment problem. It's an all-to-all alignment problem that says I've got these two sets of strings on the right-hand side. I want to figure out which strings should I try to align with which other strings. So given two individual strings on the left-hand side, like a little lowercase s and lowercase t, the alignment problem is a dynamic programming problem, which in the worst case is an order n squared computation where n is the length of the two strings. So it can be quite expensive. We use faster heuristics to cut down in the search space, but nevertheless, it's still a fairly expensive computation. And this is one of the most computationally expensive parts of the overall application. This piece of code, by the way, the string alignment is something that can be done efficiently on a GPU, and we have integrated that into MetaHitmer, but I haven't yet done I haven't showing you any performance numbers on it right now. But the right-hand side is looking at, well, now I have a set of strings, all of which I want to align against each other. So one set S might be the reads and the other set T might be the contigs. So how do I avoid calling this fairly expensive alignment kernel on every pair of strings, the order n squared, where n is now the size of those sets, on all of the pairs of things? Well, we do that by using a technique called seed and extend. We do it with k-mers. You've got a hash table. You take your strings and you put, let's say, all of your contigs, you put all of the k-mers in them into a hash table. And then for each one of the reads, you're trying to figure out what to align it to. You look up all of its k-mers in the hash table. And if you find a hit, that means there's a contig that you should align against. That may seem like a very expensive thing to do, but it still substantially cuts down on that order n squared computation. It does require a lot of communication, which still can be relatively fine-grained. The contigs are somewhat larger than the reads, and so that helps a little bit in terms of moving around larger chunks of memory. And then, as I said, not doing as many alignments as if you moved everything to everything. Now, I'll just comment here also for other genomics applications. This problem of this all-to-all alignment and string alignment comes up all over the place. It's used in multiple phases of MetaHitmer, and it's also used in comparing metagenomes to each other. It's used in a large set of things where you're trying to understand whether, in some cases, one of your sets may be a singleton. That is, you're looking at a human genome reference and you're aligning a lot of things to that. But there are many cases that come up where you're doing one of these all-to-all kind of alignments. Very expensive computation, and this data structure gives you an efficient way of getting a scalable distributed memory implementation that's also computationally efficient. So these are some scaling results from MetaHitmer. This is actually the earlier version, MetaHitmer 1. MetaHitmer 2 is actually somewhat faster, but I happen to have these across a large number of nodes for MetaHitmer 1. And this shows you the breakdown of where the contig generation, which includes both the k-mer analysis and the generation of contigs in this particular graph. That's the blue line up here, and the scaffolding, which is the last step that I didn't talk about really at all, other than to mention that it occurred. That's a graph algorithm, and is something that had been a bottleneck earlier because of the fact that there were large connected components in that contig graph, and that's been made much more efficient now in the UPC++ version. This code is available from the Exabiome website, and it is open source, and the version 2.0 was recently released. The quality, we have a separate paper looking at the quality of the assemblies. It is very comparable to other metagenome assemblers, with the exception that I mentioned that we can do much larger assemblies and therefore get better quality by doing co-assembly of these enormous environmental data sets and things like that. So just to summarize some of the features that are used in UPC++ in the MetaHitmer implementation, it uses certainly C++ templates just to make the code efficient. There's a lot of code reuse of things like hash tables and then distributed hash tables, which are built on distributed object types in UPC++. The asynchronous kind of logical all-to-all exchange where you're everybody is sending their k-mers out into into buckets of a hash table or histogram that looks like an all-to-all exchange, but is not done as a bulk collective call. It's done using these finer grained, dynamically aggregated messages or data transfers. And this gives you much better scaling relative to the MPI implementation, as I mentioned, for the k-mer counting phase. There's a future chained workflow, so using the future features of UPC++ that allows you to then get multiple RPC messages overlapped, and they're sent by the node and then by the process, so that we can get some aggregation also at the node level. So that helps also in terms of increasing our message size. And we use the promise and kind of fulfill feature of UPC++ that allows us to trade off the memory use, which with the performance, and allows us to live within a fixed size memory footprint, which is very important for the UPC++ for this MetaHitmer application, which tends to be very memory intensive. And so those are all some of the features of UPC++ that are really valuable in the MetaHitmer application. So just to wrap up, these are the resources that are available. I think you've seen these URLs before, but this gives you some ideas of where the UPC++ code and everything is available. If you are just looking at this last part of the tutorial, there's also more training material available online, the formal specification of the language and the links to the publications and things like that are all available through those websites. Here's a couple of testament from the users of UPC++. Stephen Hoffmeyer, who wrote the latest version, MetaHitmer 2.0 in UPC++, really said it's been very helpful in producing these, using these features such as RPCs for rapid prototyping. And of course, he was also the author of the COVID simulation, the lung simulation example that I mentioned as well. And another user from PNNL talks about the value of this one-sided model, and once it's written in even another one-sided model such as SHMEM, MPI, RMA, or SHMEM, that it's very easy to adapt that code to UPC++. So hopefully that gives you some ideas of why you might want to look more at UPC++, especially if you have an application that has very irregular patterns of communication, of dependencies, of synchronization, things that will make it somewhat challenging to put it into a bulk synchronous style or make it inefficient in a bulk synchronous style. I think all of those are good sort of reasons to consider using one of these global address-based languages, and specifically UPC++, which gives you the convenience of these RPCs to really move computation around to data and helps actually with some of the synchronization issues as well as just the locality issues associated with that. So thank you very much. If you've made it all the way through our tutorial, we really appreciate you spending the time with us today virtually, and hope that you will have a great experience programming in UPC++. You also can contact the UPC++ group through that website as well, the URL I have here. So good luck, and thank you very much for listening.